Hello, my name's Mark Temple, and this is a presentation of the In Brief series on cancer genetics. And in this presentation, I'll be talking about how the cells control the cell cycle, so how they control a cycle of growth. So we're looking at how cells go from a newly made daughter cell through to growing, synthesizing their DNA, growing some more, and then undergoing mitosis to give a fresh daughter cell. So one cycle of growth is referred to as the cell cycle, and the cell cycle is controlled by what we refer to as the cell cycle clock. So the cell cycle clock is a molecular circuit made up of proteins and expression of genes that operates, and the cell cycle clock is responsive to external factors such as growth factors and internal signals as well. And the cell cycle clock decides, if you want to think in those terms, the cell cycle clock decides whether to enter a new phase of growth or to retreat into a non-growing state, a quiescent, non-proliferating state referred to as G0. So, as we know, the cell cycle consists of passage from G1 through to S phase, G2, and then on to mitosis. And following mitosis, the cells may re-enter G1 for another phase of growth, or following this, the cells may enter a quiescent stage where the cells no longer proliferate and they sit there metabolizing, performing their function, but not growing. And then at some point, these quiescent cells may make the decision to re-enter the cell cycle and enter another cycle of growth. Now, these decisions are controlled by the cell cycle clock, and the cell cycle controls the movement through the phases and the decisions whether to enter or exit the cell cycle. Now, the cell cycle clock itself is responsive to other factors such as growth factors and other external signals as well. It is also responsive to internal signals within the cell. So we're going to have a look at some of these, the factors, some of the players in the cell cycle clock, and we'll also look at a tumor suppressor and how the absence of this tumor suppressor affects the cell's ability to control the growth. So what is the cell cycle? Effectively, it's a network of interacting proteins, and it can be thought of as some sort of signal processing circuit. In the way we think about circuit boards, the way they process signals and make decisions on which way to send the signal. Well, the cell cycle clock is a signal processing circuit, and it makes the decision of whether the cell's going to enter or exit the cell cycle and pass through the different phases of the cell cycle. So, because we're talking about a cell cycle clock, this is often represented in a bit like a clock face. So I'm sure you've seen this many times in the textbooks, which is one cycle of the cell growth. So here's a cell in quiescent phase, and then at some point external factors trigger the cell cycle clock to enter G1, and then during G1 the cell's growing and metabolizing and has to fulfill various prerequisites before it can then enter a stage of DNA synthesis. Following this, there's another gap phase where the cell's growing and metabolizing, creating things it needs, so that it can then enter into mitosis phase to give a daughter cell. And then the cell may either exit the cell cycle or continue in another round of growth. So clearly this kind of cycle is very important for understanding cancer, and one of the ways we think about cancer is cells that have lost their ability to regulate the cell cycle. They've lost their ability to regulate growth. And when we talk about oncogenes and tumor suppressors, typically they're important proteins that affect the running of the cell cycle and the control of the cell cycle. And later on, we'll look at the role that the retinoblastoma protein has in controlling the cell cycle. So cancer cells are cells that have lost their ability to regulate the cell cycle clock, and they're constantly in a state of growing. So the cell cycle also contains checkpoints, and these can be thought of as points of quality control, if you like. So effectively the cell is constantly monitoring itself and making sure that all of the things required before it can enter the next phase are in place. So typically there's 4 cell cycle checkpoints shown here, and in cancer cells these checkpoints often no longer function. So there's some of the cell cycle machinery required to monitor these checkpoints aren't in place, and the cell will keep growing even in the absence of even when it shouldn't be growing. So for instance, there's a checkpoint late in G1, and it's the DNA damage checkpoint, and effectively the cell won't enter synthesis of DNA if the genome is damaged. And often this is overcome in cancer cells, and even though you've got an unstable genome and a damaged genome, the cells are still growing and proliferating in an uncontrolled way. So as I was saying, cancer research often focuses on these checkpoints or transition points. Typically the entry into G1 from a quiescent stage is an important checkpoint that's been researched, and also the checkpoint in G1 prior to entering S phase is also an important checkpoint. And typically in cancer cells, they've lost their ability to regulate these checkpoints in the cell cycle. So late in G1, there's an important decision made for the cell, and this is referred to as the restriction point. So late in G1, the cell assesses itself just before this restriction point. Once the cell passes this restriction point, typically it's going to go through a full phase of growth through to re-entering G1. So it'll go through S phase, G2, M phase, and then re-enter G1 once it passes that restriction point. So it's a decision point of whether to grow or not. And once it passes this point, it's committed to a cycle of growth. Now we're going to have a look at some of the key players in the cell cycle machinery, some of the key proteins that are involved. And it probably comes as no surprise that kinase proteins are involved in making decisions of which genes are going to be switched on or off, or which proteins are going to be switched on or off to instigate cell growth. So proteins that are present in the cell can be switched on or off through post-translational modification, such as phosphorylation. And the enzymes that carry out phosphorylation are kinase enzymes. And the kinases that are involved in cell cycle clock and cell cycle growth and progression are referred to as cyclin-dependent kinases. So these are kinases whose activity is dependent on its association with a cyclin. Okay, so the CDK itself is not active, but the CDK in the presence of a cyclin is active. So we'll have a look at these cyclins and some of these cyclin-dependent kinases. So shown here is, as we're progressing through the cell cycle, is the expression level of the cyclin B protein. So the gene that codes for cyclin B is switched on, and then its expression increases through to late M phase, in which case its expression falls off. And this is for one cycle of cell growth. clearly this is occurring each time the cycle of growth occurs. The expression of this cyclin rises and falls and rises and falls. Now don't forget, this is a cyclin. This cyclin is going to associate with a cyclin-dependent kinase. And then once it associates with the kinase becomes active, and then it can turn on a whole bunch of things in the cell. And clearly there's more than one cyclin, and there's more than one cyclin-dependent kinase, and these interact in very specific ways. And shown here are a few other cyclins that are present in the cell. So we've had a look at cyclin B, whose expression peaks in M phase. There's also a cyclin D, whose expression is predominantly in G1 phase. And there's cyclin E, whose expression peaks entering S phase. And there's other cyclins as well. So there's a whole bunch of these cyclins whose gene regulation is cell cycle dependent, and therefore the associated kinase with each of these cyclins here is inactive until the cyclin levels reaches a certain level in the cell, and then that kinase becomes a dominant kinase, turning on a bunch of proteins. So cyclin-dependent kinases, the kinases themselves phosphorylate other proteins on serine or threonine residues. Now, if you remember, serine and threonine are just short aliphatic residues, amino acid residues with an OH group on the end, and it's the OH group that can be phosphorylated. The other group of kinases that we've been talking about in these presentations are the tyrosine kinase receptors, and they're a special group of kinases on the receptor membranes that phosphorylate at tyrosine residues, whereas the cyclin-dependent kinases are your more typical kinase, and they phosphorylate other proteins on serine and threonine residues. So the cyclin-dependent kinase itself is almost inactive until it associates with a cyclin, and then the cyclin CDK complex is greatly enhanced, has greatly enhanced activity. It's also thought that the cyclins help to target the cyclin-dependent kinase to certain proteins as well. So cell growth is instigated by growth factors. You add growth factors to cells and cells begin to grow. Okay, so we need to have an understanding of the signaling from the growth factor to turn on the cyclin, so that the cyclin can then activate the pre-existing cyclin-dependent kinase. So we've looked at cancer signaling in another one of these presentations, and in this presentation we're also looking at some signaling as well. And here we have a fairly unwieldy diagram from a growth factor through to activation down here in the center of one of these cyclins. Okay, and this figure is showing that there's a couple of pathways here, and there's pathways here. There's lots of different signaling pathways from different external factors that can instigate growth, and one of the ways they work is to signal through to the nucleus to turn on expression of the cyclin gene. And in this case, we're looking at cyclin D1. Okay. Now, if you look carefully here, this part of the signaling pathway is one that we've looked at in the cancer signaling presentation. So I'll just refresh our memory of that. So this is a cutaway showing one growth factor pathway leading through to activation of the D1 gene. And if you go back to the other presentation, I showed this diagram. And in this diagram, we were looking at a simple linear signaling pathway from a growth factor through to gene activation. And if you follow the players in this pathway, what you have here is a growth factor, that's shown here. We have a tyrosine kinase receptor here. And then we have two players that are involved in signaling through to the RAS molecule. So we have the two players, the GRB2, the SOS, that are involved in signaling to RAS. And then downstream of RAS, we have this series of 3 proteins that form the MAP kinase pathway. So that was the MAP kinase pathway is here. It's RAF, MEK, and ERK. And then when we were discussing this, we then just said that the final kinase in the MAP kinase pathway phosphorylates some transcription factors to turn on a bunch of proteins, newly synthesized proteins, that are required for a change in phenotype. Now, we're looking at a specific example of this here. And in this example here, we're looking at how the ERK protein here is able to switch on the D1 cyclin gene. And the phenotypic change that results from this is progress through S phase of the cell cycle. And the new bits to this pathway that we've already looked at are some of these transcriptional activators here, so that ERK can lead through to the phosphorylation and activation of the FOS gene. And the FOS and another gene called, or another protein called JUN, work together to form the AP1 transcription factor. So downstream of the MAP signaling pathway, we have some phosphorylation events leading to the phosphorylation and activation of FOS and JUN, otherwise known as AP1. Then AP1 binds to the promoter region of the cyclin D1, and then cyclin D1 is expressed. The CDK associated with cyclin D1 becomes active, and then it can turn on a bunch of proteins and help the cell progress through the cell cycle. So now we've got a bit of an understanding of how external factors can signal through these pathways that we've already described, leading to gene activation. And one of the genes that's activated, particularly in a cell cycle dependent way, in G1 is the cyclin D gene. So now we're going to look at one of the well-studied tumor suppressors, which is the retinoblastoma protein, or the PRB or RB. So RB can refer to as the gene, RB can refer to the protein. Mutations in the retinoblastoma protein have been identified in the cancer retinoblastoma as well. So the retinoblastoma protein is a tumor suppressor, and tumor suppressors, as we've already discussed in one of these presentations, their presence in the cell can inhibit growth, and then the cell can temporarily inactivate these genes to allow growth, and then they become active again to halt growth. So effectively, tumor suppressors act a little bit like a brake on a car. And we've also discussed oncogenes, and oncogenes effectively drive growth rather than suppress growth. So oncogenes act a little bit like an accelerator on a car. And in cancers, both tumor suppressors and oncogenes tend to be perturbed, and this leads to uncontrolled cell proliferation. So we'll have a little bit of a look at the role of the retinoblastoma protein and how it is involved in regulating the cell cycle. So in this diagram, the extent of phosphorylation of the retinoblastoma protein is shown as you progress through the cell cycle. So the cell cycle is shown here, as we've seen before, as this kind of clock face. And outside of this, shown in red here, is the phosphorylation status of the retinoblastoma protein. So in early G1 phase, there's very few phosphate groups on the retinoblastoma protein. So it's referred to as hypophosphorylated. Okay, and then at some point in late G1 that coincides with restriction point, retinoblastoma protein becomes hyperphosphorylated. It picks up phosphate groups and at many of its serine and threonine residues, and this phosphorylation increases through to the end of mitosis, whereby it's heavily dephosphorylated back to its hypophosphorylated state. So you can see here the phosphorylation status of retinoblastoma is cell cycle regulated, and clearly the extent of phosphorylation affects the function of the protein. Okay, so when retinoblastoma acts as a growth suppressor, so in G1, when it's not phosphorylated, it's suppressing growth. And then when it becomes hyperphosphorylated, it stops suppressing growth. And in cancer cells, in the absence of retinoblastoma, effectively you have no halt on growth. So in cancer cells, you don't have functional retinoblastoma. But in normal cells, as shown in this cycle here, you have a functional retinoblastoma during early and mid G1 phase. And once retinoblastoma becomes hyperphosphorylated, that's the trigger at restriction point, allowing cells to undergo a cycle of growth. So retinoblastoma is often referred to as the guardian of the restriction point, and when it becomes hyperphosphorylated, it loses its growth inhibitory function, and it allows cells to progress through late G1 and then enter a complete cycle of growth. Now, question is, how does the retinoblastoma protein have an effect? So the presence of retinoblastoma, when it's hypophosphorylated, when it's not phosphorylated, retinoblastoma binds to a transcription factor or a family of transcription factors referred to as E2F, or the E2F family of transcription factors. And when retinoblastoma is hyperphosphorylated, it dissociates from these transcription factors. And then these transcription factors, in the absence of retinoblastoma, turn on genes required for DNA synthesis and therefore allow the cells to enter S phase. So this is a schematic just repeating what I was saying. So in early G1 phase, retinoblastoma protein is hypophosphorylated, so it's lightly phosphorylated, and in this state it binds to and sequesters the E2F family of transcription factors. Through the action of cyclin-dependent kinases that phosphorylate retinoblastoma, so one of those being cyclin D, that we know that cyclin D levels build up during G1, activates its associated cyclin-dependent kinase, and then it phosphorylates the retinoblastoma. And once retinoblastoma is heavily phosphorylated, it breaks away from these E2F transcription factors, leading to active E2F factors. And then these active transcription factors bind at the promoter region of genes to drive the DNA synthesis phase. And the promoter region of many genes involved in DNA synthesis and DNA metabolism are those that contain the binding sites to the E2F transcription factor. And then at some later stage in S phase, the E2F proteins are sequestered or broken down. Even though retinoblastoma is still heavily phosphorylated, there's other cellular controls that regulate and remove the E2F transcription factors. So the E2F transcription factors are momentarily active as the cells cross restriction point, moving into, preparing the cell so that it can move into S phase. So shown here are the E2F transcription factors with a dimerization partner here, bound at the promoter region of a gene. And this is in the absence of retinoblastoma. So you could, if you like, you can say, well, this is what it's like in a cancer cell. You don't have the retinoblastoma protein regulating these E2F transcription factors, and you've got these genes are turned on. But in normal cells, what happens is that the E2F transcription factor, whether it's free in the nucleus or bound to the DNA, is not active, particularly when it's bound to a hypophosphorylated retinoblastoma protein. So the retinoblastoma is stopping the E2F responding to growth signals, and it's stopping it from transcribing genes. There's clearly it's a bit more complicated than just an interaction with retinoblastoma, and one of the other players that's heavily involved in this are the histone deacetylase and the histone acetyltransferase. So this shows the histone deacetylase, and we've looked at the deacetylases before in another one of these presentations. It was the one on tumor suppressors. So in this situation here, the histone deacetylase is tethered to the transcription factor complex, and when you bring the deacetylase, it removes the acetyl groups from the nucleosomes and causes the nucleosome to bind tightly to the DNA, and this represses gene activation. So the retinoblastoma, it binds to E2F, and another way in which it represses gene expression is by tethering in the histone deacetylase, which makes the DNA more compacted and less easy to transcribe. Conversely, in the absence of retinoblastoma, the E2F protein is able to tether a histone acetyltransferase or a histone acetylase. So these proteins, they add acetyl groups to the nucleosome cores. And when you add the acetylases, you change the charge of the histone tails, if you like, and this loosens up the nucleosome and allows it to be transcribed. So I think I've already mentioned this, but which sort of genes are then transcribed by these E2F factors? They're genes involved in synthesizing DNA. So there's a whole bunch of genes involved in synthesis of DNA, whether it be making nucleotides or making some of the enzymes required for actually copying the DNA. And at the promoter region of these genes, if you do your bioinformatics and you do your studies, you can see there's binding sites at these promoter regions for the E2F transcription factors. Hence, they're able to affect these genes specifically. So hopefully that's given you a bit of an understanding of how a tumor suppressor can suppress growth by sequestering a transcription factor, and also gives you a bit of an understanding of how a growth factor can signal through to turn on a cyclin gene in a particular phase of the cell cycle to control the cell progression through the cell cycle. Okay, thank you for listening.